Okay. Good morning, good afternoon, and good evening to everyone joining around the world. Welcome to the Global COVID Lab meeting. My name is Sabrina Welsh, and I'm the Director of Programs and Operations at the Human Vaccines Project, and I'll be your moderator for this webinar. The COVID-19 pandemic continues to expand, with over 17 million cases confirmed globally, with 4.4 million of those cases recorded in the US. According to the WHO, there are now 25 vaccine candidates currently in clinical testing, and just this week on Monday, we saw two major phase 3 clinical trials begin on the same day. Both Moderna and Pfizer and BioNTech announced the beginning of their phase 3 trials, each of which will enroll 30,000 people in the United States, and later the Pfizer and BioNTech study will enroll participants globally. Additional phase 3 studies are ongoing or set to start later in the year, so it's going to be really interesting to see these clinical trials progress and generate data in the coming months. Today, we are convening to bring you the latest information from experts across the field in a forum open to scientists globally. To date, more than 1,400 people have registered for the Global COVID Lab meeting, and we're so pleased that you're joining us today. Just a note before we begin, the information presented today includes some pre-published data that is currently under peer review. At the request of the author, we ask that you not reproduce or disseminate the data presented. This session will be recorded and available on our website and social media channels for you to review later on. I'm very excited to have Dr. Galit Alter as our speaker today. Dr. Alter is a professor of medicine at the Reagan Institute of MGH, MIT, and Harvard. Her research is focused on the development of systems biology tools to define the correlates of immunity against infectious diseases that ravage the globe. Her work points to unexpected mechanisms of protection against HIV, malaria, and tuberculosis, has led to the development of novel diagnostics to monitor chronic infections and diseases, and now promises to accelerate the development of novel classes of therapeutics able to deploy the activity of the innate immune system in a specific and controlled manner. In today's lab meeting, Dr. Alter is going to talk about her work using systems serology to define antibody responses to SARS-CoV-2. During the presentation, please send me your questions using the Q&A function in Zoom. I'll collect your questions and ask her a broad selection of your questions after she's finished her presentation. We'll have about 25 minutes for discussion, and I hope you'll take the opportunity to ask questions on the data presented today. It is now my pleasure to introduce Dr. Galit Alter from the Reagan Institute of MGH, MIT, and Harvard.
Thanks,
So
Sabrina.
if you want to share your screen.
Yes. Okie dokie. So thanks so much, Sabrina, and thank you to the HVP for inviting me to be a part of this lab meeting. And what I thought I would show today is some new preliminary data that we've been compiling over the last few months, aimed at defining the correlates of immunity of natural SARS-CoV-2 infection, with the hope that this might illuminate how we believe antibodies might provide protection against this virus in the context of vaccination. So before I begin, any talk about SARS-CoV-2, I have to really give credit to the folks out there in the world doing this creative work of developing memes and jokes to help alleviate some of the somber feelings I think we have as we've been trapped in our homes for months upon months. And this is one of my favorite memes that I just love to share with the community out there, just really to just give, put out the point that, we've been developing antibody tests and we've developed incredibly robust tools now to track and to really understand where the virus has gone over the last few months within different populations. But we still don't really know what those antibodies mean. So a long time ago, I feel like, time moves at an incredible pace in this particular pandemic. Back in March, we were looking very, just broadly at antibody responses across different populations of individuals that were coming to the hospitals and treating and testing positive in the RNA tests. And what we began to appreciate, as many others have published in the field, is the fact that there is this incredible heterogeneity in antibody responses across individuals who were once infected. And interestingly, this heterogeneity is shown here both for IgG titers and convalescence, IgA titers and IgM titers. These are all to the receptor binding domain. And they really don't necessarily track always solely based on the severity of disease. But we would see is that there were some individuals that had very high IgG levels, others had quite low levels, and the same was true for the other isotypes. And so this really got us scratching our heads, really trying to get us to start thinking about how we could begin to understand what it was about an antibody profile that could in some way provide information about how that individual interacted with a virus once they first were infected, and how that might predict whether or not that individual was going to have severe disease, moderate disease, or perhaps even help them resist a pathological course of infection. So we began to think about how we could apply our system serology tools that we had been developing over many years to look at immune responses to multiple other pathogens, but now to really begin to apply these to COVID-19 patients and to really begin to look deeply at the humoral immune responses in individuals that were testing positive once they were admitted to the hospital, and to begin to think a little bit more clearly and a little bit more deeply about what it meant to have a COVID-19 immune passport. What is the flavor of antibodies that we think will truly give us information about whether somebody is resistant or protected from infection? So the one thing that I think became clear from multiple discussions across the globe is that antibody results, antibody tests, would tell us that somebody was exposed, but it didn't necessarily tell us that that humoral immune response was intimately involved in the response to the virus and whether or not it was actually involved in immunity. So what we began to do was to think really about how we could redeploy our tools to begin to understand what flavors of these antibodies might be most vital for protection. And so where the field has moved, quite logically is towards this idea that individuals who raise antibodies are able to bind to the virus in just the right way, in a way that allows them to essentially block de novo infection, so to elicit neutralizing antibodies, is really key to the induction of antibodies that will be critically important for preventing infection. And clearly we see through most of the vaccine studies that have now emerged in the public domain is that vaccines that can elicit neutralizing antibodies can effectively prevent viremia at some level in the upper and lower respiratory tract. And so that's really quite exciting. Now, the confusing thing about these neutralizing antibodies is that what we saw in infected individuals, differently than what we were seeing in vaccine studies, was that individuals that had the most severe infection were those that had the highest levels of neutralizing antibodies. So we began to ask ourselves if it's not only neutralization that matters for the humoral immune response that might be critical for containing a pathogen that has already caused an infection. Are there other features of that humoral immune response that might in some way play some role in the containment and clearance of the pathogen? So of course, for all those who've heard me present before, what we are particularly interested in is the role of antibodies in their ability to interact with the immune system. And so beyond their ability to simply bind and block infection, antibodies play a critical role in providing very specific instructions to the immune system, and specifically to the innate immune system, to destroy anything to which those antibodies are bound to. And so beyond just binding, once these antibodies are stuck to the surface of an infected cell or to the virus itself, these antibodies can recruit a wide array of different forms of innate immune functions, including the ability to secrete cytokines, recruit phagocytosis, drive NK cell cytotoxicity. They can drive degranulation. They can lead to complement deposition. They can even prime more effective adaptive immune responses. And so really trying to understand specifically whether there were antibodies elicited in some populations that were infected with SARS-CoV-2 that were able to elicit particular innate immune functions, and if those were somehow related to different kinds of clinical outcomes, as well as to understand what was specifically different about these antibodies, we felt could have important implications for both monoclonal therapeutic and vaccine development. So what we set out to do essentially was to begin to readapt all of our tools that we have developed for system serology to now begin to profile the SARS-CoV-2 response across multiple different types of sample inputs. We began with first looking to see if we could adapt the tools to look at population level differences in antibody responses to the virus. We also looked in developing tools that would allow us to model different forms of monoclonal antibodies and how they might interact with the viral antigens and then deploy different forms of innate immune function, as well as adapt all of our tools not only to look at non-human primates that are obviously a critical bridge animal model for vaccine studies, but also to begin to look across different small animal models because of their importance in helping us understand at a single cell level and a host pathogen interaction level exactly how this virus might interact with the immune system to drive different forms of pathological or protective outcomes. We took plasma or antibodies from these different source inputs. And then what we do typically is profile these responses in two different ways. We perform biophysical profiling on the antigen-specific antibodies on the top, and we also perform functional antibody profiling on the bottom. For the biophysical profiling on polyclonal pools of antibodies from humans or animal models, we initially look at the different subclass isotype distribution across up to 500 different antigens derived from that pathogen of interest. And so these can include peptides or epitope, knockout variants, or different antigens from the virus, as well as different SARS-CoV viral antigens. And we also purify these antigen-specific antibodies, and we ask if they interact with different Fc receptors or complement with different affinities. And we also purify the antigen-specific antibodies from these particular individuals, and we ask if they are glycosylated in different ways, because we know that the sugars attached to the constant domain of antibodies are so critical for deploying different forms of immune effector functions. Now, in parallel to this biophysical profiling, we also ask if the antigen-specific antibodies from our pathogen of interest can fix complement, activate neutrophils, activate macrophages, monocytes, NK cells, eosinophils, dendritic cells, if they fix antigens more effectively in germinal centers, as well as if they can activate other cells of the immune system, those from the adaptive arm, as well as how they interact also with other cells like epithelial or endothelial cells, which are a critical target, at least for this particular pathogen. And we take all this data collectively together and we integrate the data, and we use machine learning approaches to essentially find patterns in the data that allow us to understand the flavors of immunity that might track with particular populations of interest. So let me give you an example of how we applied this. So I'm going to tell you two stories today that will give you an idea of how we took system serology and applied it to SARS-CoV-2 infection to begin to understand how antibodies might contribute to the resolution of infection. So what we did in the first story that I'm going to tell you about is we took a cross-sectional look in acute SARS-CoV-2 infection. We applied this technology and we asked, what is the difference in individuals with different clinical trajectories? At the end of this story, it raised many questions about what it was about that early response that led to these very different clinical outcomes, and I'll show you that in just a minute. And then so the second story I'm going to tell you about is how we then looked over time in individuals. In that first month of interaction following symptoms between the host and the pathogen to begin to understand how the humoral immune response evolves in individuals who are able to conquer or clear the virus. So the first story is really quite simple. And essentially what we did is very early on in February, we partnered with Helen Chu at Harborview Hospital in Seattle, really at the center of that first outbreak in the US. Helen had collected samples from acutely infected individuals that were hospitalized at the time of hospitalization. And so essentially she captured all of these individual samples between 5 and 10 days following symptom onset. So they were all sampled very early on. Now, the individuals were then followed over time, and 12 of the individuals went on to convalesce, and 10 of the individuals unfortunately went on and passed away. And what we're doing is we're sampling very early on, really at this one single time point at the time of hospitalization, and we asked whether or not there was any flavor of humoral immunity very early on that could predict different outcomes, these two different clinical trajectories. So what I'm showing you here is the first data from this analysis. And so the way to look at these particular graphs, the way to interpret these graphs is, I'm going to show you over here on the left side for IgG1 levels. So we measured the levels of antibodies to the spike antigen, so the whole spike antigen, the receptor binding domain of the SARS-CoV-2 virus, as well as to the nucleocapsid antigen from the virus. And the way to read this here is each of these distributions represents one of the two populations. The blue distribution represents the convalescents, and the brown distribution over here represents the antibody profiles in individuals who went on to pass away. Now remember again, everyone was sampled very early on essentially at almost the same time point following symptom onset. And now we're going to look at these response profiles across the 3 major isotypes that we measure in early infection. So hopefully what you can see here from the distribution is that they're relatively similar across the different antigens as well as across the 2 individual groups. And really after Bonferroni correction, really for statistical rigor, we found that there were no real profile univariate differences across these 2 groups that could really discriminate individuals that were going to go on to convalesce or go on to pass away. There were some interesting kind of clues here that potentially there were, lower distributions of IgGs and IgMs, particularly individuals who went on to pass away, particularly for spike. But there was nothing that really, really stuck out that told us that this is really why there are differences in these individuals' response to the pathogen. So we were interested now in essentially taking all the data we had collected from system serology and ask if it's not the absolute titers of these antibodies, is there a quality shift, some sort of interaction between these profiles that are elicited early on in infection that could give us some clues about differences in how antibodies might be acting as biomarkers of different treatment outcome or different clinical outcomes, or might even tell us about their direct role in antiviral immunity. So what I'm showing you over here now is the aggregate data for the humoral profiles that we captured for these individuals. And the way to read these particular graphs is each radar plot represents the response in a given population. The top are the deceased, the bottom are the convalescents. And what we have here in the columns are the different antigens that we profiled. So spike, receptor binding domain, or nucleocapsid protein. And the way to read these particular radars is depicted over here, where on the left side in the bluey purple petals in these flowers, these are the different subclasses and isotypes of antibodies to these particular antigens. And in the yellowy orange on the left side of these radars or flower plots shown over here in these particular petals, these are the different functions that we captured for the same antigens. And so these include monocyte phagocytosis, neutrophil phagocytosis, complement deposition, as well as NK cell activating properties of these antibodies. And the size of the petal tells you how strong that response was in that given population. So hopefully I can draw your attention to the first set of petals or the first set of flowers on the right side here. And these are the responses to the nucleocapsid protein. And what we found, and hopefully you see this as well, is that the size of this radar or this flower was bigger in the individuals who went on to pass away compared to the individuals who ended up convalescing. So this was really quite striking. That was true for both the subclass isotype distribution as well as for particular functions, with a significant expansion of nucleocapsid-specific antibodies able to elicit phagocytic and complement depositing activities. These were expanded 5 to 10 days following infection, specifically in individuals who went on to pass away. Now, conversely, if you look all the way to the left and we look at the spike-specific response, the profiles were different. This time, the flower was a little bit more disjointed, but the overall structure of the flower was larger in the individuals who went on to convalesce compared to those who went on to pass away. What was striking about this particular flower is that there were really these larger leaves of IgG1s, IgAs, and IgMs in these individuals. So on an overall level, their magnitude was higher. And this was linked to higher levels of spike-specific monocyte phagocytosis, neutrophil phagocytosis, and complement activation. Now, interestingly, there were 3 petals that were larger in the individuals who went on to pass away, and these were related to spike-specific NK cell activating antibodies that were interestingly enriched in the deceased, not so much in the individuals who went on to convalesce. Now, the one confusing part about this particular analysis, and I do want to draw attention to this, I think this is really important potentially for the discussion, was the receptor binding domain response here listed in the middle. Here what you can see is that the receptor binding domain antibody response was bigger in individuals who went on to pass away compared to those who went on to convalesce. And interestingly, we automatically think that antibodies to the receptor binding domain are likely to be highly critical for protection from infection, at least in the context of vaccination. But what it looks like is in the context of natural infection, it's really these antibodies that accrue in individuals who are going down the wrong path as opposed to a path that might be related to containment and control of infection. And we can discuss more about why that might be. But the spike-specific response looked really quite interesting. Big flowers, particular functions associated with convalescence. So to begin to understand the relationships of these functions, because we were really curious about why it was that NK cells were not popping up in these individuals, we assumed, at least in the context of flu, that ADCC is protective. So we thought that ADCC would also be critically important in the context of SARS-CoV-2. So we began to look at the relationships of all of these features to begin to understand what was different early on between individuals who survived versus those who passed away. So what I'm showing you over here is a correlation matrix. And the way to read these graphs is really quite simple. Every measurement we made is listed on the left side, and the exact same measurements are made along the x-axis. And so what we're asking here is, if I make high levels of spike-specific IgG1s, do I also make high levels, for example, here of RBD-specific IgG4? So you're looking at the coordination of any two given features within that humoral immune response. Where there is red in the plot, it means it's highly correlated. Where it's blue, it's anti-correlated. And so what we're looking at here is the level of red to blue, and we're looking at where that red to blue falls within these plots, particularly looking at comparing the convalescents to the deceased to begin to understand the differences in the flavors of immunity that might be related to that early response to the virus that is protective. So there are two interesting profiles I want to point out here. The first is this little blue strip in the bottom of the convalescents, which is actually very red in the bottom of the deceased. And this little blue strip relates to the coordination of the evolution of neutralizing antibodies with all other antibody qualities and functions. And what's interesting is in the convalescents, they don't coordinate neutralization with any other antibody function. So what it looks like is these individuals make an immune response. They elect to have lots of different functions in their antibodies, including phagocytosis, NK activation, as well as complement deposition, but they don't coordinate it with neutralization. Neutralization may not be essential in this early contained response to the virus. Conversely, in the individuals who go on to pass away, they might still be making immune responses, and they make these neutralizing antibodies, and these neutralizing antibodies are coming up in concert with the evolution of the overall humoral immune response. So very different coordination of the Fab and the Fc domain of the antibody. Now there's another really striking difference between these correlation matrices. On the right side over here in the deceased, you have a big blue block that is not present in the convalescents. And if you look a little bit more deeply at what is found in this big blue block, you see that this is nearly all NK cell functions. So what it looks like in the deceased individuals is they make pretty strong NK cell NK activating antibodies, or they make phagocytic antibodies, but they never coordinate these two functions together. They don't seem to elicit this more polyfunctional clearing activity with this cytotoxic activity. They really seem to induce the either or, but never bring them in together. Whereas the convalescents that may not have had really high levels of ADCC, what these individuals do is they bring in ADCC with phagocytic and complement activity. And so this gave us clues that maybe that ADCC may be important, but it's important in the context of a clearing or opsonophagocytic response that is essential very early on to eliminate the infection from the lung compartment. So we were fascinated by these multivariate differences that we began to see here. So we elected next to go into a larger multivariate model that didn't just look at two-way correlations, but look at the relationship of all the features together to begin to understand if there was a minimal signature of antibodies that was elicited very early on in infection that could explain different clinical outcomes. So what I'm showing you over on the left side is the outcome of this multivariate analysis. So we took all the data that we had captured. Initially, we applied a feature down selection to eliminate any possibility of overfitting. And we took those antibody features and we asked, could we resolve individuals who were convalescent from those who ultimately passed away, as you can see here on the left side. We had almost complete separation, almost no overlap between the antibody profiles in these individuals. So simply by capturing this antibody profile information, one could determine very early on whether someone was going down a deleterious clinical course or if they were on their way to potential recovery. Now, interestingly, here in the VIP scores, I'm showing you the features that were selected by the model to drive this separation, and really only 5 of the total 70 features that we had captured were required to separate out these 2 groups. And the 5 features are listed over here based on their importance to separating out individuals within the model. It's not necessarily their magnitude of difference, it's just how important they were to separating out the 2 groups within this model. The features that are marked here in blue were enriched in the convalescents, and the features marked in brown were enriched in the individuals who went on to pass away. And hopefully what you can see here, very interestingly, is that there was a very striking split here, where all the blue features that were essential were directed towards the spike antigens for IgM and IgA titers, whereas all the features that were associated with ultimate death were all specific to the nucleocapsid antigen, including the ability of nucleocapsid-specific antibodies to drive complement deposition, as well as titers of IgMs and IgAs to the nucleocapsid. And those 5 features was really all you needed to separate out the 2 groups from one another. Now, because the model really is trying to get rid of all the overcorrelated nature of the humoral immune responses induced in these individuals, we next want to go back and look to see what other features are associated with these particular biomarkers that might give us additional insights, immunological insights, into why it was that these features were enriched individuals went on to convalesce, and these features went on to ultimately pass away. So what I'm showing you over here on the right side is the correlation network of all the additional features that we had measured that were correlated very highly to these particular 5 biomarkers of disease outcome. So down here, the 2 big blue dots are those that were associated with convalescence. This was the spike-specific IgA levels, and here are the spike-specific IgM levels. And what was fascinating is if we look at the features that were tied very closely to these particular biomarkers, these were related to spike-specific neutrophil phagocytosis, spike-specific monocyte phagocytosis, multiple antigen binding to the Fc receptor involved in phagocytic activity, as well as the ability of RBD-specific antibodies to fix complement. So what this told us, really interestingly, was that it might not have been the IgA and IgM titers alone that were driving the outcome, but that these particular biomarkers are tied to particular functions, to spike and RBD, that are critically important for driving uptake and clearance of the pathogen through this opsonophagocytic mechanism of potential viral clearance. Interestingly, over here in the gray dots are the features that were highly upregulated in the individuals who went on to pass away, and these were highly tied to multiple nucleocapsid humoral immune responses, as well as the subclasses with less function. So what this told us is that there's just really this divergent immune response to S and to N, so spike versus nucleocapsid, and the spike-specific responses that are essential for this prediction of convalescence was really associated with these highly functional responses that might be essential for the clearance of the virus. So the way that we began to interpret the data and we began to really think about it was that maybe what these biomarkers are telling us is that S is good, which is not surprising, and that making responses to nucleocapsids with the humoral immune response might be derailing the immunodominance of the immune response. So what we set out to ask next was really whether or not this ratio of spike to nucleocapsid immunity was a marker of early evolutionary differences in the protective immune response. So first, to prove to ourselves that this profile that we were seeing in this cohort in Seattle was really reproducible, we next went to a second cohort, a validation cohort. And so what I'm showing you over here in the original Seattle population is this S to N ratio for all the functions that we had measured. And so hopefully what you can see here is if you look at the overall ratio of spike-specific functions in convalescence versus the deceased, you can see this very clear difference in this immunodominance profile, in the humoral immune response, in the individuals in the Seattle cohort that I had just shown you. But interestingly, the same immunodominance profile was present in the first 10 days following symptoms in a second validation cohort from Boston, really where the significance here was even higher. It's not completely resolvable, but it gives us incredibly important clues about the ultimate trajectory these individuals were going to take. But now focusing on these individuals, we began to ask a little bit of a different question. And the question was really, is it really that S is higher than N at this very early time point? Or is there some sort of longitudinal trajectory that changes between S and N that might in some way inform whether or not maybe these individuals who passed away just were, a little slower in making that response, or maybe they're just really immunodominantly different and they're never able to make a full spike response. And maybe that's why they ultimately pass away. So to really gain some information about what was different in their trajectory over time. So to address this question, in the Boston cohort, here what we did is we recruited samples from 85 hospitalized individuals with moderate infection, 72 individuals in the yellow here that had severe infection were admitted to the ICU, and then 33 individuals who went on to pass away. And these individuals, again, were recruited 5 to 10 days following symptom onset, and these individuals were followed over the first month of their infection. And so what I'm going to show you now is really what those trajectories look like to begin to understand if this S to N variation early on is really a fixed phenomenon and the deceased never evolved immune responses, or if there's something simply different about the trajectory of these responses that tells us about the essential immune responses that must be in place early on in infection to control the virus. So just to initially just show you what these trajectories look like, I'm showing you here the spike-specific IgG response and the nucleocapsid IgG response in individuals who were severe but convalesced, here in the yellow, versus individuals who were severe in burgundy that were also admitted to the ICU but unfortunately passed away. And hopefully what you can see here is that what is different about these individuals very early on, these individuals who unfortunately pass away, is that they do have a significantly lower spike-specific response early on, which might ultimately lead to this different ratio of S to N very early on. But they do try to make these responses. And interestingly, by the time they're unfortunately, close to death, they do catch up in some cases in some of these responses. But there's some sort of very interesting early defect in their response that seems in some way to prevent them from fully leveraging the immune response to fight the disease. Interestingly, we saw no difference in the nucleocapsid IgG response between these two individuals. But let me just show you now how deeply we went into these humoral profiles and really where all the differences lie in their immune responses to begin to really understand what is special about this evolving humoral immune response that is associated with different clinical outcomes. So I'm going to compare the blues, the yellows, and the burgundies for you, and we're going to look initially at what this looks like across the isotypes. So first, looking at the IgM response to spike, what I'm showing you here is the response at the first week, second week, and third week following symptom onset across the 3 different groups. And hopefully what you can see here is for the IgM response, there really are no differences really in their response to the virus. And there's a little bit lower in the moderately diseased individuals here in the blue, but really the severe individuals, whether they survive or don't survive, seem to have similar IgM responses. Next, looking at the IgA responses, it's very similar. The burgundy and the yellow are very similar over time to one another. The blues are a little bit lower. They have moderate disease. Potentially, they have less virus in the system, or maybe were had a more effective early control, barrier control against the virus. But the IgG response was interesting and was different. So if you look here at the burgundy distribution, the blue distribution, very early on in the first week, the IgG response to S is really low. It's much higher in individuals who go on to survive infection with severe disease. Interestingly, as time goes on, the yellows continue to evolve. The blues really have this remarkable evolution of IgGs, but these individuals who pass away really have this diminished evolutionary response in the context of IgG1 responses to spike. And the same is true for the IgG3s, where we see here this remarkable trajectory of IgG3s in the individuals who go on to survive, and we see a diminished trajectory of evolution in the context of individuals who ultimately pass away. So we became interested in other features of these antibodies. How do they interact with FC receptors, for example, because we think that this clearance mechanism is so important based on that early data out of Seattle. And so we began to look at the ability of antibodies to interact with the FC alpha receptors. These are IgA receptors on different immune cells, as well as the FC gamma receptors, FcR2a involved in activating phagocytosis, 2b, which is an inhibitory FC receptor, FC gamma R3a involved in ADCC activity, as well as FC gamma R3b involved in neutrophil binding. And hopefully what you can see here is very similar to the trajectories we had seen with IgGs. We see here that the individuals who survive tend to have higher level responses, particularly at week 2 following symptom onset, compared to the individuals with moderate and severe disease who pass away. And this difference is sustained early on, although some of these antibodies do seem to try to catch up. But the major difference really occurs at 2 weeks, where at 2 weeks, these guys just do it better. So while these individuals who pass away have equal amounts of IgMs and IgAs, they really fail to induce their IgGs early, and they fail to leverage their functional activity, which is really quite peculiar. To confirm that at a functional level, we now measure the ability of these spike-specific antibodies to mediate monocyte phagocytosis, neutrophil phagocytosis, complement deposition, and K-cell activation, as well as to neutralize the virus. And what we found systematically across all these functions is that very early on, the survivors have higher levels of each of these functions. By week 2, they are profoundly higher. This is really the critical time where you see these massive functional antibodies in the individuals who survive that do not seem to come up as high in individuals who ultimately pass away. And interestingly, they do catch up by the 3rd week, but that seems to be too late. If you look at the neutralizing antibody response, it really follows the same trajectory, maybe a little higher in the people who pass away very early on. But then you see that these individuals who survive make these higher quality antibodies. And this is true both for the FC functions as well as the FAB functions that are just really expanded individuals who are able to survive infection. So we can look at this in an aggregate way here. We're looking at these flower plots again, severe individuals who survive on top, individuals who are severe but who pass away on the bottom. And again, we're looking here at week 1, week 2, and week 3 following infection. Now, the way to read these particular plots is in the green are all the isotypes and subclasses. In the pinky purples, we have all the FC receptors. And in the orangey yellows, we have all the functions. And hopefully what you can see if we look at the severes, you see this beautiful evolution over time. These are the people who convalesce. They just evolve in all their humoral immune responses over time, and they make these beautiful balanced, really functional immune responses that are highly effective in this context of this particular infection. In individuals who pass away, they have these punctated, very strange responses where they have decent isotype and subclasses, but their FC receptor binding is really just, not very good early on. And then you can see that there's a stunted evolution of function in these individuals where they do end up evolving these isotypes and subclasses, but they don't involve the same level of functionality and FC receptor that is present in these individuals. And again, you see that this flower is somehow squished in these individuals by the 3rd week of infection. So there's really this interesting overall humoral defect in the evolution of these responses. So we want to make sure that this evolutionary defect is not related only to age. So here, just looking at the distribution of age across all the samples based on this multivariate profile that we captured. And you can see this interdigitation of beige and red all throughout. So age was not what was driving this antibody profile difference, nor was gender. Here, the reds are females and the blues are boys. And you can see that it's not that there are really, it's a gender difference in the antibody profile. The major difference in the antibody profile is shown over here, where you have many more of the individuals who pass away in the burgundy down here compared to up here, where you see all individuals who survive. So the major difference in the antibody profiles is not age or sex driven. It is really driven by these different flavors of immunity that evolve over time in these individuals. And just to look at them as single smooth lines, here are just some examples of what the profiles look like in the individuals who survive in the yellow versus those who pass away in the burgundy. And you see this beautiful trajectory in the individuals who survive. And then you see these kind of more droopy types of evolutions in individuals who go on to pass away. And here I just kind of outlined what these types of humoral immune responses look like. But what we did note here was that there was both a droopiness at the end, but there was also a delay early on. And so that got us thinking that there's a little bit more going on with these immune profiles than simply just that they're decaying faster. So we began to look at trajectories of curves. We started applying different kinds of mathematical models to begin to understand really how and where these differences lie. And so here we asked, well, which features are most different between the survivors and the non-survivors? And here we have a list of these features where those on the left side, where we have the biggest delta AIC, are most different across these two groups. And if you now look at what these curves look like down here, what you can see here is for Fc gamma R2b binding by S2 specific antibodies, these individuals here who survive have these responses very early on. They're already present at the time of symptom onset, which is really fascinating. And then these individuals who pass away ultimately, really take their time evolving these responses. And then when they come in, they just basically bounce up, but they might just be there too late. Whereas other features that are popping up over here, like the Fc gamma R2a S1 specific response, that just really takes an awful lot of time to evolve individuals who ultimately pass away. And so we're seeing is interesting immune dynamic differences, not only across the S-specific response, but it's fascinating to see that it's also different across the different components of the spike antigen, telling us that antibodies to different regions might also contribute to convalescence, but they do it through the leveraging of different kinds of immune functions. So everyone always asks, well, what's different between individuals with moderate disease and those with severe disease? Well, I can tell you the differences in their humoral profiles are huge. Most of them are really with respect to the magnitude of the response. But what you see here is that the major difference is really always in leveraging Fc function rather than titers alone. So they really are utilizing their antibody Fc interactions in a very different way in severe infection compared to in a moderate infection. And here you can see these trajectory differences where these individuals who have severe disease really evolve these responses early and they really have them at higher levels. In the moderate infection, I don't think this is any evidence of enhancing disease, really, because we don't see that there's ever any evidence of particular antibody functions that are associated with ultimate passing away. But it is an interesting observation that there really are these differences, very big differences between the moderate and those individuals who go on to survive infection. And then finally, comparing the moderate and the deceased, because they do have a diminished response compared to the severes, the differences are not very different, which is really interesting between these two groups. But there are features that can discriminate these two groups. The differences are not huge, but they are significant if they're above the black line. And interestingly, if we look at what is most different across the individuals, these two groups, it's always that individuals who ultimately pass away make higher levels, particularly of IgAs in response to infection. And these are across multiple different parts of the viral antigens, primarily S2, S1, as well as S. And these really come up more in individuals who pass away compared to those with moderate infection. Although we also do see evidence that the individuals who pass away also make antibodies, are able to leverage Fc receptors more aggressively than we see individuals with moderate disease. So what I just want to leave you with is this idea that these differences are really striking across both the severe individuals who survive as well as those who pass away. Really, this is related to these different trajectories of humoral immune responses, as well as we see this in individuals who are severe, the moderates, and those who end up passing away. But although their responses are more similar, these blues and purples, really what we see is that there are some fundamental differences in how these individuals who are severely infected, who have this blunted humoral immune response, they do seem to leverage more IgA. They might gain those responses, but they may not be sufficient to provide protection and ultimate convalescence following infection. So hopefully what I can leave you with is this idea that early COVID-2 responses really do help to differentiate and delineate who is going down different clinical trajectories, and it points to particular functional responses that might be critical for protective immunity. And that really it's all about this early trajectory of the humoral immune response that points to this idea that these individuals who convalesce are really leveraging highly functional antibodies that might be directly contributing to the control and clearance of the pathogen. And we to date do not see any evidence of enhancing antibodies in the context of our particular patient population. And with that, I just want to thank the people who did the work in Seattle. Helen was really on the front line, got us samples very early on. The group here in Boston, Rochelle and Ed, who got us samples in Boston. The team here on the computational and lab side who did all the work, as well as all of our funders. And I'm sorry, I went a little long.
Galit, that was fantastic. Thank you so much. we are getting literally dozens of questions. So I'm going to try to get through as many of these as we can. I just wanted to check before we start the Q&A session. Is it okay if we go a couple of minutes past the hour? Because we were getting some really interesting questions. Okay, thank you so much. I think I'll let you keep your slides up in case you want to refer to some of your data, because you had some really interesting data slides, and a lot of the questions are coming in. You may want to refer back to them. First question, could the differences in those who contain virus versus those who continue to progress be related to viral load and persistent immune stimulation and inflammation? And are there any cellular data to compare and integrate into the analysis, including CD4 and CD8?
Yeah, so that's a really good question. So we did do, so we had viral loads on the initial Seattle cohort, and when we stratified based on viral loads, there was no difference. That's, viral loads that we were seeing in the oropharyngeal swabs, as well as, we actually had viral loads also from the blood, and we couldn't see anything there. That doesn't mean the viral load is not different in the lung, right? And so I really do think that what we're seeing here is some sort of biomarker of more virus in the lung in the severely infected individuals that ultimately pass away. So I think that, there is no question that viremia, I think compartmentalized viremia might definitely be different across the groups. For the second population here, in the Boston population, we do have some T-cell data. It's not quite as extensive as we'd like. And interestingly, in our hands, the T-cell responses are equally higher in severely infected people compared to those who are moderately infected. So T cells and antibodies seem to be moving together. And there are differences in the T cell profiles. They seem to be less exhausted in the individuals who have moderate infection. But there's no T cell profile so far that we've seen that is really as predictive as these antibody biomarker differences. That doesn't mean the T cells are not more important in the lung, but at least peripherally, the antibodies seem to be more predictive of differences.
Okay, interesting, great. Okay, moving on. The difference in epitope reactivity among survivors versus deceased is striking. Even more striking is the difference in type of FC activity elicited by survivors versus deceased. Could you comment, do we know if this is a correlation or causation? Meaning if some people die who have an underlying condition, is that inspired by the incorrect target or FC type? Or has the incorrect target or FC type itself led to the poor outcome? It's an interesting question. Also, can we separate people with underlying conditions who instead survived?
Okay, so that's a really big question. So I think the studies are not big enough to know if there are polymorphisms that are going to pop up. So far, I have not seen any of the GWAS data that suggests that there's any particular polymorphism in FCRs that is associated with different outcomes. I think that what we're seeing here mostly is that individuals are just leveraging their humoral immune response in different ways to different targets. I think, what's interesting here, I think that the question asker is getting at is really that there seems to be this early decision that the immune system is making to either leverage these FC profiles or not towards one target or another. It looks like everyone's getting these inflammatory antibodies. They're incredibly effective, but they're going towards a good target or a bad target. My inclination, and this is really just something that we have no data yet to really support this, is that these nucleocapsid protein specific immune complexes are probably just driving inflammation in the lung. They're probably leveraging complement, they're probably driving complement pathology, and this might be going down a deleterious path, whereas these particular antibodies that are able to hit the virus and the infected cells might be able to clear those immune complexes that have infectious particles, but these are not really infectious particles, so they're just driving non-specific inflammation. So I think that might be part of what's happening in that context. And then finally, to get to the causal question, there are not just us, there are multiple groups that are doing FC modifications to monoclonal antibodies to begin to ask how FC contributes to outcome. And I think that those are going to come in the very near future.
Nice. Yeah, that's good to know. Okay, did you look at similar antibody profiles to any of the 4 primary coronaviruses that cause the common cold in these same patients at various stages in their hospitalizations? And are they responding to the COVID virus based on their baseline antibodies to other coronaviruses?
No evidence of cross-reactivity. It's wild in our data.
That's the question, yeah.
Yeah, so we have only receptor binding domains from the 4 common coronaviruses. So the NL63, OC43, HKU1, and 229. We don't have the full spike antigen, so it could be that we're just not looking at the full cross-reactive antibody population, but we see zero evidence of any baseline differences in common coronavirus immunity and the overall evolution ultimately of the CoV-2 specific response, or that there's an expansion of those responses in these individuals. They look pretty they look pretty like locked in and steady. They don't seem to be evolving in the context of these infections.
Okay. Since you focused on clearance rather than prophylaxis, can you predict which therapeutic convalescent plasma or neutralizing monoclonal antibody preps will be better or worse for therapy?
Oh yeah, so that's like, I guess, the golden question, right? So I think so I my bias is I think that I have not really seen any convincing evidence yet that there's authentic enhancement in COVID-2 infection. I know that there is this data from SARS-CoV-1, and of course there's a lot of other viruses where something's going on, but it does seem to me that having some function on board may be critical in order to get complete clearance and protection against the virus. My feeling is, and this is again another one of my gut instincts as opposed to data that supports this, is this idea that if a virus escapes the nose, even in the context of prophylaxis, if one virus escapes and goes down into the lung, at that point it's going to be in infected cells most likely, and so neutralization is just not going to be enough. And the only way then to get rid of that particular infected cell or that small propagating infection is really to leverage immunity. And so whether that's T-cell immunity that can kick in and do this, or if it's an FC-enhanced antibody that can do it, I think we need that backup strategy. Without that, we're basically relying strictly on having sufficient titers in the upper nasal that's going to give enough of a barrier for protection. So I really think that there is a need for some immune recruitment, whether it's T-cell or innate immune through antibodies.
Sure, yeah. We've received a couple of questions about the demographics of the volunteers enrolled. Was there any impact of age or other demographics obvious? And I'm curious also because the Human Vaccines Project is very interested in the aging and elderly immune responses and how the effective age might change the immune response to the virus and treatment outcomes. So did you see any age effects?
Yeah, so what I'm showing you over here is the distribution of ages across the entire population based on the antibody profiles. And so what you can see here is that the dark reds are individuals who are over 80, individuals here in the beige are under 40. And what you can see here is it's not statistically significant differences in the distribution, although there are a few more dark reds kind of located down here, and you can see this is where the severes live. But overall, I mean, there obviously is an age-dependent effect. It's a clear comorbidity associated with negative outcomes in SARS-CoV-2 infections. There's no question something going on. In our population, it was not biased in that way. But what I can tell you is that we have now, going into even larger populations, we see very clearly that, there are age-dependent defects in how we leverage FC function in our antibodies. And this may have larger implications, not just for SARS-CoV-2, but it could have, influence on response to flu, to RSV, to many other pathogens that we don't deal with quite as effectively in the context of age.
Okay, we're receiving a couple of questions about RBD and the M proteins. Let's start with this. Could you further comment on the role of RBD-specific antibodies, especially IgG and IgM, on the survival of participants?
Oh yeah, so this weird plot, I don't know, I would love somebody to tell me what this means. So I think that, okay, this is the way that I interpret this data. So, but this is really, up for discussion. So I think that what we see is that neutralizing antibodies increase with severity of disease and tend to be highest in individuals who unfortunately often pass away. And so I think that what this means is that, if you happen to make a strong enough S-specific response early on that is able to clear the virus sufficiently, you may not need to continue to evolve your response to more specific epitopes within the viral envelope. So I think that what this is this is not, this data does not indicate that RBD is a bad target. It does not indicate that RBD specific responses are, associated with enhanced infection. I think what this suggests is that these individuals have continued evolution epitope spreading towards other regions of the SARS envelope, and that if you happen to have this just might be enough, and you don't need to make more, because otherwise we wouldn't see this effect here, which is really quite profound to me, right? Your neutralization does not continue to evolve in the context of these individuals who convalesce. So they might just have enough immunity early on to spike that they're clearing and getting rid of the virus, and then they can contain, as opposed to these guys that just keep seeing virus and it just takes them down the wrong path.
It's fascinating, the difference, and see that type of correlation. I haven't seen that before, so that's really interesting. Going back to the questions now about M proteins, are there correlations of patient outcomes with antibodies against M and E proteins? And did you look at responses to membrane proteins in convalescent and deceased individuals?
So we've not looked at it in deep enough, really as deeply as we should. At the beginning of the outbreak, back in February and March, we didn't have access to M&E. More recently, we have put M&E in. The responses are weaker to M&E, at least in our hands, but it might also be that the antigens are not really quite as high quality. And so if there is anybody out there that has good M&E antigens and are interested in working together, we would be delighted to do that. But so far, we haven't seen anything striking yet in the Boston, this larger longitudinal cohort with M and E, but we do have them in the analysis.
Okay, and this may be a moot point based on your answer just now, but are there antibodies against M protein contributing to ADE in these individuals, or is that not observed because we don't know quite yet?
Yeah, so it's possible, right? Anything is possible. I can't say definitively. I would assume that there is a lot more M on the surface and E, so that it is possible that they could be derailing the response in a negative way. But I assume the spike-specific response and the RBD-specific response is also quite abundant, and it's really quite high titer. And it's not that so severes have more because they have more antigen in the system. So just like in tuberculosis, just like in malaria, if you have more pathogen in the system, you're going to make more antibodies. That doesn't mean that those antibodies are enhancing. If you look in the people who go on to pass away, there's no single antibody profile feature that is enhanced in those individuals that points to true enhancement. That doesn't exist in any of the populations we've looked at. It may be possible for M&E that it's there, but at least for the antigens we've looked at so far with S1, S2, trimeric forms, monomeric forms, anti-deleted spike, everything we've looked at so far, we have not seen anything that really suggests that there's something like bad that antibodies are doing.
Okay. Is the fact that IgA is higher in deceased patients suggesting that gut symptoms are a deleterious sign for the course of the infection?
Oh, that's an interesting thing to look at, to think about. I did not think of it that way. The way that I interpreted the IgA data is that these individuals clearly have a burst of viremia in their lung. They have very high levels of viral loads, and if you look at their NP swab levels, are equivalent to those of the severe people who end up surviving infection. The way that I interpret it is that they have this massive burst of IgAs and IgMs. They just don't class switch to IgGs. And so what that kind of drew our attention to in the public, in like sort of historical publications, is that there is considerable levels of IgA class switch that's CD4 T cell independent. And so maybe these people are making these very early bursts of IgMs and IgAs because there's so much antigen in the lung, but they're not getting the T cell help to push them towards a protective IgG response. And maybe that's why we see lots of IgA, but no IgG early on. That would be what I was thinking, but maybe it is also associated with different other mucosal symptoms.
Sure. So how do we put these findings in the context of vaccines? What responses should we look for in a potentially effective vaccine?
Yeah, that's like that's the really interesting question. So I think that some of the work we've done already with Dan Baruch's group looking at both DNA vaccination as well as Ad26 vaccination work that's just come out today, we clearly see a role for Fc effector function as a co-correlate of protection in vaccination. So neutralization is really critical, but the Fc functions are also coming up in line with neutralization in animals that have the highest degree of protection. Where it becomes really interesting in all of these animal model studies with vaccines, and I think what's going to come out also in the humans, is that it may be that strict neutralization, again, will be associated with protection in the nose. But Fc effector functions may be more relevant once you get down deeper into the lungs. And so what I'm really curious about is how we begin to develop correlates analyses in our animal modeling for all these different vaccines that will allow us to understand what does it take to completely block infection up here, and what is it going to take to prevent disease as we go down further into the lung, in case some people might get bigger boluses of exposure than others.
That's the $64,000 question, I think, for sure. More on the...
Yeah, go ahead.
No, go ahead. I was going to say.
I was going to say that so far we have not profiled any vaccine that has not really pointed to phagocytosis and complement being essential. It's really fascinating.
Okay, and more on the topic of vaccines and their responses, will you have access to the antibody responses in the Moderna vaccinees to look at their profiles as you described? And same question for all the different vaccine candidates out there.
Yeah. So we'd love to. We are in discussions with Barney and Bob and Mario at the VRC and Andrea at Moderna to potentially also profile the responses with the mRNA vaccine. That would be really exciting, and I'm sure we will be involved at some point, whether it's for this paper that just came out on Tuesday or whether it's the upcoming studies that are ongoing. We'll figure that out for sure. We have already been working with Novavax, which has been really quite thrilling, and they've got a beautiful response. And we are embedded in the Merck program and also discussing possibilities with other groups as well. So we're very excited about participating, and with GSK, I should mention as well. They've been really wonderful and forthcoming.
Super, that's great to hear. And we've actually got Dan Brook as our next speaker in the series. So that was well timed. I'm sure he'll be focusing on the paper came out today. His paper
today, this morning.
Fantastic. Okay, haven't seen it because we're doing this, but I'll take a look. It'll be great to hear his thoughts on that as well. Okay, so many more questions. Let's do a couple more minutes if that's okay.
Sure.
Let's see. And we're receiving a lot of compliments just how clear and interesting this data is on immune responses. I think it's one of the most clear presentations on immune responses to SARS-CoV-2 that we've seen. So thank you so much. Okay. Have you compared the antibody profiles to systemic inflammatory, so cytokine and chemokine responses? Do patients with early high-quality antibodies have less systemic inflammation since they can presumably clear the virus better compared to those with delayed or less effective antibodies that fail to control the infection and thus develop excessive tissue-damaging inflammation?
Okay, so you cannot completely unlink those, right? That is a really critical point because clearly the comorbidities associated with outcome, right, are age, BMI, and some of these other like diabetes and other types of comorbidities that are associated with inflammatory changes. So one of the major questions that we're trying to understand from a pure population level pathology standpoint is how are those populations of individuals leveraging their humoral immune response in different ways because they have this different inflammatory background that may predispose them to not making these highly aggressive IgG responses. Mm-hmm. That is absolutely a possibility that is a complete unknown. But I think that once we begin to understand this, once we can unlock the mysteries of how those predisposing comorbidities influence our ability to leverage the humoral immune response, I think we'll be able to come up with better vaccine strategies that can overcome those limitations, right? That is the key, is if we can figure out why they don't do it. Is it because they have too much TGF-beta or IL-10 or something else early on that prevents this? I think that we'll be able to come up with strategies to try to overcome this. With this little thought in the back of our heads that we don't want to over-exaggerate that immune response and potentially drive at enhancing function. That's going to be absolutely critical to think about that balancing outcome.
Yeah. Relatedly, we've received a question about functional responses. Most of the vaccine, the phase 1 and 2 trials seem to mainly be looking at titers of neutralizing antibodies. And your research seems to show that it's key to look at functional responses. Do you think that will be part of the phase 3 trials? I know a lot of the vaccine developers are talking about outcomes and the importance of, what do we look at as the cutoff? What is the relevant outcome that we should be looking for in efficacy trials? So could you comment on that a bit, please?
Yeah, so titers and neutralization are key, right? There's no question that we can, look away and not prioritize those as key metrics for vaccine take and vaccine efficacy. But I would find it very surprising if FC will not be part of the discussion, because I think there are quite a few individuals discussing the role of FC. That was clearly a major consideration in design of monoclonal therapeutics that are going into populations. It is also something that I hear brewing in the context of the convalescent serum transfer studies. And so I would be shocked if in the end, there are not going to be some FC readouts, whether they're going to be primary outcome measurements, right, Serena, or whether they're going to be secondary, profiles that are going to be captured to understand the efficacy of trials. I think that's what, still has to be decided. But I think that, more and more, I think as you'll hopefully see in some of these in Dan's studies where we've done this deep dive into the antibodies, these FC profiles just provide just that extra little information that helps us understand how the FAB and the FC can be fully leveraged in order to capture that virus and really prevent it from causing disease. And it was just wild in the AD26 study because he's got one arm that has like no nasal or lung viremia. It's just a wild arm and it has this burst and beautiful FC profile that just really makes it really quite striking how important that combination of FAB and FC work together.
So interesting. Okay, just a couple more quick ones. I think, did you correlate the responses with HLA types of the patients?
Not in the full cohort. So we have HLA typing for about 100 individuals in this cohort, and we haven't yet seen, I mean, it's too small to know definitively if there's anything there. We haven't seen any particular class 2 that pops up as being really strikingly different. And I think that we're not powered to really see class 1 effects, to be completely honest, because we can do the super types and CD4s. So I think that, you know, eventually I think that, you know, as we can do this on larger populations of individuals, we'll be able to really pull it out. But I'm convinced that there is a CD4 trigger early on that is pushing us down the right path or not. And that CD4 response, I think, is really what is key to driving these, you know, highly effective immune responses.
Okay, I think we're going to wrap up so we don't take too much of your time. Dr. Altel, thank you so much just for sharing your insight and for presenting this data. You know, it's so valuable for the community to get to speak with scientists such as yourself who are on the front lines. And thank you so much for your time. I know you're very busy, so we really appreciate your time. Thank you.
Thank you.
I'm going to go ahead and take the screen back, share a couple more slides with upcoming information on our next talk. Okay, so I invite you to join us 2 weeks from today on the next Global COVID Lab meeting, which is scheduled on August 13th at 10 a.m. Eastern Daylight Time. Our next speaker will be Dr. Dan Baruch from the Beth Israel Deaconess Medical Center, who will talk about COVID-19 vaccine development and a bunch of studies published by his lab. He had 2 papers in Science back in May and now just one today, talking about exposure to the disease and administration of vaccine conferring protective immunity to SARS-CoV-2, which is hailed as extremely good news by scientists for vaccine development. And if you're interested in more research on COVID-19, please sign up for the HVP COVID report. In this newsletter, we present insights from experts around the globe and highlight the latest scientific articles and data. And finally, please feel free to visit our website and follow us on LinkedIn, where we'll upload a recording of this webinar for your review. Stay safe, wear a mask, and we hope you'll join us next week, in 2 weeks, on the next Global COVID Lab meeting. Thank you so much. And Dr. Alter, have a great day. Thank you.
Thanks, you too, Sabrina.
Bye.